Today, we are very happy to have two distinguished speakers to give the seminar. Let me first introduce Dr. ZHAO, our first speaker. Dr. ZHAO Xin, hello.
Hello, can you see me? Can you hear me?
Yes, we can hear you. Yes, thank you. Dr. ZHAO is Associate Professor at the Department of Biomedical Engineering, Hong Kong PolyU. Her research interest is on translational regenerative medicine and working in multidisciplinary area. Her area cover material science, cell biology, engineering, medicine, etc. And aim at cell in microenvironments for various medical applications. So far, she has published 100 articles, and she was listed as one of the highly cited research year 2022, a very prestigious nomination. And she has successfully attracted over 20 grants and published in very top journals. So here the floor is yours. We are so glad to have you as our speaker. Dr. ZHAO, please.
Yeah, so you probably need to stop the screen sharing.
Yeah.
Stop. Yeah, okay. Great. So hello everyone. So today we are going to talk about the photo-crosslinkable biomaterials for tissue regeneration. So this is like the kind of my major research interest in my research group now. My group focus on three directions. So one thing is we based on different needs, we synthesize different materials. So what we mainly focus on the materials is the photo-crosslinkable polymers. And then we make these we do the scaffold fabrication. So we make these kind of materials into microspheres, nanofibers, or injectable, or 3D do some 3D printing as well. And then we apply these materials into to regenerate tissues. So mainly like bone and skin and also tendon. And also we do a bit of the cancer therapy as well. So here is like the reason why we actually focus on the photo-crosslinkable materials. So photo-crosslinkable materials, so we see that this is a monomer. So initially these materials, they are liquid. And upon this, and this is the crosslinker. So upon the light exposure, so these the crosslinkers can join together, and then we're going to form this kind of cross-linked polymer network. And by controlling this polymer network, we can control the mechanical and also the degradation properties of the materials. And also because the materials is initially liquid, so what we can do is for example, we can inject them through a needle and then like using the 3D printing, so we can inject them through the needle and then print and also the crosslink layer by layer, and then we can create these 3D scaffolds. And also because they are initially liquid, so we can pour them into some very fine molds, and then in the beginning they are liquid, so you can fill into different shapes, and then we crosslink them and they will be solidified, and then we can peel off these from the mold, so and then we can form these kind of fine patterned structure on top of the materials. So today we are going to talk about two types of the photo-crosslinkable polymer. One is the synthetic polymer, and also is the other one is the natural polymer. So this is a kind of cartoon showing the how we can form these kind of three-dimensional photo-crosslinkable materials. So as we can see from this cartoon, we can know that so by joining these monomers together, so we can form a three-dimensional network. So and also we can, by controlling the cross-linking density, so we can control the properties of the materials. So we are first going to talk about the how we can use the photo-crosslinkable synthetic photo-crosslinkable materials synthesized from our lab. And then one thing we are going to talk about is how we can use them as a 3D printed scaffold, and also how we can micro pattern them for bone and also bone osteogenesis and also the angiogenesis. So here we know that because the materials, they are initially liquid, so we can inject them through the needle, and then we can print them layer by layer. And then by cross-linking them, we can form this kind of three-dimensional network. And normally, if you're using, the reason why we want to use the photo-cross-linkable materials is because if we're using other polymers, so either we need to use the high temperature to melt them, to make them like liquid, or we might need to use the organic solvent to dissolve this polymer. In this case, we won't be able to load the bioactive molecules inside the materials. And it's because the photo-cross-linkable monomers, they are initially liquid. So in this case, we can encapsulate these bioactive molecules inside the materials, and then we can print them and make these kind of 3D scaffolds. And here is summarized like the major photo-cross-linkable materials. So we can have the natural polymer, for example, methyl acrylate alginate, gelatin methyl acrylate. So because these are normally hydrogels, so they might have the good biocompatibility, but their mechanical properties may be low. And also we have the synthetic photo-cross-linkable materials, for example, polyhema and also polymethyl acrylate. But these materials, normally they are not degradable. And also when they cross-link, so when they solidify, they might generate a lot of heat. In this case, we won't be able to encapsulate the bioactive molecules. And also we have the composite materials. For example, for bone regeneration, a lot of times we're using the hydroxyapatite because they are chemically very similar to bone. But the problem is like when we are actually mixing them together, so we might have the ceramic aggregation, and then the interaction between the polymer and also ceramic is weak. So in this case, we will sacrifice the mechanical properties. So what we do is like, this is our project aim. So we want to develop a photo-cross-linkable 3D printable composite materials with excellent mechanical, biological, and biomolecule releasing properties. So this is the materials we synthesize. So we have so we also like this is one of our major research interests in my lab. So we have the propylene glycol to make it injectable, and also the lactide to make it degradable, and also the methyl acrylate to make it photo-cross-linkable. And also by varying these kind of N and M, so we can control these cross-linking density, and then we can control the mechanical properties. So now we can actually make these materials very soft and also very elastic. And also we can make these materials very strong as well. And the other component we put is the methyl acrylated hydroxyapatite. So in this case, these materials, because they have the ZHAO bond C there, so they can cross-link either with themselves, and also they can cross-link with the polymer. And then they can form these organic-inorganic double network. And then these will strengthen the mechanical properties. And also hydroxyapatite, they can buffer the degradation products during the polymer degradation. And the reason why we actually put the ZHAO bond C to the hydroxyapatite, so we think that if we are using the naked hydroxyapatite nanoparticles, so when they are mixed with the polymer, we are going to form a composite like this. Whereas if we are actually making the hairy hydroxyapatite, so what they do is that they can cross-link with themselves, and they can also cross-link with the polymer. And this will increase the mechanical properties. So this is just to show you some data. We proved that we successfully synthesized the polymer. And this is to show you we successfully synthesized the hydroxyapatite nanoparticles. So for each like the each hydroxyapatite nanoparticles, so we calculate that there are about like 700 to 800 these kind of brushes. And this is something important. So we can see that, so this is C1C plus hydroxyapatite nanoparticles. So this is the naked hydroxyapatite nanoparticles. So if we don't cross-link them, we just mix them with the organic solvent, we can see that for the C1C misacrylated hydroxyapatite nanoparticles, so after the 12 very long time incubation with the organic solvent, because the interaction between the nanoparticles are very big, so these can actually increase the colloidal stability of the nanoparticles, so they won't sediment to the bottom. Whereas if we don't have the brushes there, so these nanoparticles, they will assembly, and then they will sediment to the bottom of the organic solvents. And also, if we use the UV light to cross-link the materials, we can see that if we use the materials can be self-cross-linked. So when we use the UV light, so because the hairy hydroxyapatite nanoparticles, they can cross-link with each other, and they become larger nanoparticles, and they will sediment to the bottom. Whereas the naked one, they won't cross-link. So this will show that these kind of adding the C1C to the hydroxyapatite nanoparticles, they will increase the colloidal stability of the hydroxyapatite nanoparticles. So in this case, we can mix these very well with the polymer, and also they can cross-link with each other. And then this will also strengthen the mechanical properties of the composite. And then we show the examine the photo-polymerization rate. So we can see that adding these hydroxyapatite nanoparticles, they won't actually they don't have the detrimental effect on the cross-linking rate, and the composite can be fully cross-linked within about like 140 seconds. And also, like by using the different MMNs, and also changing the concentration of the polymer and also the hydroxyapatite nanoparticles, and also changing these kind of how much hair we add to the hydroxyapatite nanoparticles, we can greatly change the mechanical properties of the polymer. And we can actually make these materials within the compressive modulus range of the trabecular bone. And this is also something very important. So conventionally, when we are using these kind of photo-crosslinkable polymer, a lot of times we are using the HEMA. So if we cross-link the HEMA, we can see that the temperature will increase to about 70 to 80 degrees C. Whereas when we are actually cross-linking our polymer or composite, the temperature will only increase to about like 36 degrees. So this is advantages is because if we want to encapsulate the bioactive molecules like growth factors into the materials, so because the materials will not go up to the high temperature, so they can preserve the bioactivity of the materials. And we also encapsulate using the materials to encapsulate the bone morphogenic protein too, and then we release them. And we find that these the materials composite can encapsulate and also long-term release of the bioactive molecules. And also because we want to 3D print these materials, we also examine its viscosity. And we find that by changing different polymer ratio and also the polymer and nanomaterial concentration, and we can control the viscosity. And we are also able to print these kind of the 3D scaffolds with high resolution. So nowadays, so this is like some paper published like about 2 or 3 years ago. So nowadays we are able to manage to make these the resolution about 100 micron. And also we examine the degradation of the materials. And we also find that, so when we add these kind of hydroxyapatite into the polymer, so degradation product of the hydroxyapatite can buffer the degradation products of the polymer. And this is also advantageous. It's because like if we, during the polymer degradation, we might create some acid, and this acid may cause inflammation. And if we can buffer these kind of degradation products, so we can make these kind of reduce the acid, so we can reduce the possibility of the inflammation. And then we examine the biocompatibility of the materials. So these are the cells seated, mesenchymal stem cells seated on top of the materials, and we can see the green dots represent the living cells, and the red dots represent the dead cells. So we can see that most of the cells on top of the materials, they are alive, and also they can adhere and spread well on top of the materials. And also the materials can support the proliferation of the cells seated on top of the materials. And then because we use the materials for bone regeneration, we also see that if the materials can support the osteogenesis, and we use two markers. One is the alkaline phosphatase, and the other one is the alizarin red. And this ALP is the early marker of the osteogenesis. And we can see that our composite materials, they can support the angiogenesis. This is the data of the other method. So it's the alizarin red. So alizarin red state the calcium deposit. So we also see that when we are our cells on top of the composite, they can secrete a lot of these kind of calcium modules. This means that they will have better potential of the mineralization. And then we implant these 3D scaffolds into the rabbit femur condyle defect model. So we see that these green things, they are the scaffolds, and the red things, they are newly regenerated bone. And we can see that our composite materials with the BMP2, so with the growth factor, they can support the optimum osteogenesis. So this is just to show you that these are PML and DMA materials. They also by changing the different formulation like M and N, and also the concentration of the hydroxy apatite and the polymer, we can change the mechanical properties of the materials. And also the materials, they allows encapsulation and long-term release of the bioactive molecules. And also we can 3D print these materials. And then we use the same materials to use the polymer to make the micro pattern scaffolds as the artificial periosteum. The reason why we are actually making the periosteum is like one function of the periosteum is to recruit different cell types. And a lot of times, if we just implant the bone scaffolds into a bone defect, so because the cell interaction of the implant and the bone is not very good, sometimes the efficiency of the bone regeneration might be compromised. And periosteum can actually help the increase the efficacy of the bone implants. So this is the current artificial periosteum. So most of them are made of like cell sheet and also the porcine intestinal mucosa, and also some of them are made of the hydrogel. One thing is like if you look at the figure, so these are normally the artificial periosteum. They are normally mechanically not strong enough, and also they are lack of the mimicking structure of the natural periosteum. So what we do is like we try to make these kind of periosteum, which can mimic the structure and also the function of the natural periosteum. So this is our strategy. So what we use is the same polymer. So because they are initially liquid, and then we can solidify. So what we do is like we make them like on the one side, we have this kind of like a lot of brushes. And on the other side, we have a lot of channels. So for these channels, what they can do is like they can regulate the cell alignment, and then they can regulate the osteogenesis and angiogenesis at the same time. And also for these brushes, because they can increase these interactions between the materials and the surrounding tissues, so we can increase the tissue adhesion. And on top of this one, we also like put some polydopamine on top of that. So in this case, we can further increase the tissue adhesion of the materials to the surrounding tissues. So this is again just to show that we successfully synthesize the materials, and also we successfully synthesize the polydopamine. And here is how we can make these micro patterned periosteum. So we have the PDMA mold here, and we also have the liquid polymer here. And then we put the polymer between the molds, and then upon the UV exposure, we can solidify the materials. And after removing this mold, we can form this kind of genus periosteum. And then we put the materials into the dopamine materials, and then we form this polydopamine coating. So in this case, we have this adhesive genus periosteum. So on the one side, we have these kind of small brushes, and on the other side, we have different these grooves. And this is to use to regulate the cell alignment and regulate the cell fate. And this is just to show that because we have these kind of brushes, so we can increase the tissue adhesion of the material to the bone. And also we can see that the materials itself and also the materials with different concentrations of the polydopamine, they can support the cell survival, and also they can support the cell adhesion, and also they can support the cell proliferation. And then we see the different cells. One is the mesenchymal stem cells on top of the different channels. So we can see that when we are having the channels very tight and this the very tiny channels, very narrow channels, so we can actually make the cells align better. And then we are going to have the increased alkaline phosphatase and also the OCN. So these two are both the osteogenesis marker. So it means that when we are making the channels very tight, we can align the cells better, and then we can increase the osteogenesis. And then we see the endothelial cells on top of the materials. And again, we can see that when we are making the channels tiny, we can make the cells align better. And also we have the increased VEGF-A and eNOS. So both enzymes means that enhanced angiogenesis. So in this case, we can conclude that by using these different size of the microchannels, we can regulate the cell fate of mesenchymal stem cells and also the endothelial cells. And then we can regulate the osteogenesis and angiogenesis in vitro. And then what we do is like we make these materials, apply these materials in the rabbit variable defect model, and to see that how these materials can support the bone regeneration. And we can see that when we are having the microgrooved materials, and also we are having these kind of brushless materials, we can enhance the bone regeneration. And in addition to enhancing the bone regeneration, we can also see that these red circles they suggest the blood vessel formation. So these materials can also enhance the angiogenesis, so they can support more blood vessel formation. So to conclude, so we have proved that our polymer, they can be fabricated into sophisticated structure, so we can regulate the cell fate, and also we can give them modify the other physical properties like tissue adhesiveness. And by varying the cell fate, we can have the well-coordinated angiogenesis and osteogenesis. And we further using the same materials to give them some other patterns. And this is mainly for to accelerate the angiogenesis. And we know that the angiogenesis is very important for the tissue regeneration. So here's how we make. So what we do is like these is the materials. And on top of the materials, we make these living mimicking channels. So by using these living mimicking channels, we can they mimic the running track of the cells, so they can guide the vasculature formation. So this is again how we can make that. So here is the living mold. So and then we pour the materials because they are initially liquid, and they can be molded into this silicon wafer mold. And upon light exposure, the materials will be solidified. And after we remove these materials from the mold, we can form these micro patterned materials. And this is how the materials looks like. So we can see that these materials, they are very very soft, and also they can have these kind of micro patterns on top of the materials. And then what we do is like we see the endothelial cells onto the materials, and we can see that when we are using these kind of this is the flat materials, we can see that when we are having the flat materials, so the materials the cells are big and flat. But when we are having these channels, we can make these the cells, the morphology become more like pointed. So when we have like more pointed these kind of endothelial cells, they are more like tip cell morphology. So it means that they are going to more tend to form these blood vessels, and also they more tend to form these blood vessels, and also the junctions between the cells are going to be like increased. And also we again seeing these also the markers showing the blood vessel formation. So again, we can see that when we're having these kind of leafing structured patterns, so these patterns, they can guide the cell fate and make them like to have the accelerate angiogenesis. And then we put these materials again in the rat cavarial defect model. So and then we examine the blood vessel formation. And these the hemosiderin, that means that the blood vessel is leaky. So when we don't see these brown color or these kind of blue color things, that means that our blood vessels, they're not leaky, they're intact. And also we can see from these images, it's like when we're having the leafing, so the blood vessels we formed through this kind of leafing structure, the blood vessels are more intact and they won't leak these kind of hemosiderin. So this is again to show that our materials, we can make them into a sophisticated structure. So we can use this structure to guide the cell fate, and also we can use them to form these kind of more mature vascularization. And then we are going to talk about some photo-crosslinkable natural materials. So these summarize the what's the current natural photo-crosslinkable polymer. For example, we have the photo-crosslinkable hyaluronic acid. We also have the photo-crosslinkable alginate. But these two materials, they need the RGD modification for cell spreading. And also we have the photo-crosslinkable collagen. But the problem is like when we are trying to make the collagen more photo-crosslinkable, currently the method is difficult to control. And all these materials, when we want to use the light to crosslink them, it will take a very long time. And in this case, it won't be good for the cell encapsulation. And what we develop is like, and these materials have been developed many years. So what we do is like we use the C-bond-C to modify the gelatin, and the materials will have the gelatin backbone. So this is the to make it cell adhesive and also biodegradable. And also they have the C-bond-C here to make them like photo-crosslinkable. So and also the major advantage of these materials is like we can control their cross-linking density. So by many different ways. For example, we can control how much like C-bond-C to replace the amine group, and we can control the material concentration. So we can control the cross-linking density, and then we can control the physical properties like mechanical properties. So we're using these the materials like what we call JAMA. We making them like into different formats. Like for example, in situ cross-linking hydrogels, Electrospun fibers, and also we're making these cell-laden microspheres. So what we do is like, because these materials, they are initially liquid, and then they can be injected and fill into the skin defects, and upon light exposure, they will be solidified. And also because they're initially liquid, so they can fill into defects of different shapes. And then so after we crosslink them, they will be solidified. And because we have the gelatin backbone there, the materials will will gradually degrade, and then tissue will be regenerated. So this will show that how robust we can control the mechanical properties of the material by simply just changing the material concentration from 5 to 20%. We can actually tune the modulus from few kilopascal to few hundred kilopascal, and also the degradation can be changed from few days to few months. And then we examine the biological properties of the materials. So what we do is like we see the keratinocyte on top of the materials, and we can see that most of the cells they are alive. As you can see that most of the cells are green cells, and also we can see that the cells can adhere well on top of the materials. And also we can see that we form a very nice cell monolayer, and also the cells exhibit these very tight junctions. So this is something very important if we want to regenerate the multi-layered epidermis. So once we see these cell morphology, so what we do is like we lift these materials into air-liquid interface, so induce the formation of the multi-layered epidermis. So this is like after 6 weeks of the induction of the multi-layered epidermis. So we can see that. So this is the human skin, and this is the current gold standard. We use the collagen to regenerate this artificial skin, and this is what we do is like we use in JAMA. We can see that first of all, after 6 weeks of the culture, the collagen already gone, whereas the JAMA is still there. So this is what we think is very the advantages is because if we have very large wounds, so if we use the JAMA, there's no need for the second application. And also if we can see that, so the structure and thickness of the regenerated epidermis on top of JAMA collagen, they're all similar to the natural human skin. And also then we examine the function of the reconstructed epidermis on top of the hydrogel. So this is the epidermis, what it looks like, the epidermis on top of the hydrogel. We can see that once the hydrogel are covered by with the epidermis, they have the increased resistance, and also they can prevent the water loss. So it means that our reconstructed epidermis have some kind of functions, some barrier functions of the natural epidermis. So this is just to show that, so these Photo-crosslinkable hydrogel, they have tunable physical properties, and also because they are initially liquid and they can be solidified, so we can use them as an in situ forming hydrogel for wound healing. And then we make the same materials and make them like into the nanofiber scaffolds. So the reason why we actually make using the nanofiber scaffolds is because like they are like this, so they have large pore size, so these will allow the nutrition and waste diffusion, and also they have the large surface to air surface area, so that they can allow the better material and cell interactions. The problem is that when we are making the wound dressing, we normally make these materials very thick, and then once we make the nanofibers very thick, we might actually make the pore size tiny. And in this case, if we are sealing cells on top of the scaffolds, the cell may not be able to penetrate and infiltrate the whole full thickness of the materials. So what we want to do is that we want to make something soft and elastic, so we can see the cells on top of the materials, and the cells can be easily infiltrate the whole scaffold. So again, we are using the materials like JAMA. And the reason why we want to use them is because we can control the cross-linking density, and then we can control the mechanical properties of the scaffolds. So what we can do is like this is a JAMA solution, and then we electrospun them, and then we cross-link them, and then we can have these kind of cross-linked JAMA nanofibers. And by controlling the cross-linking density of the JAMA, we can control the mechanical properties of each single fiber, and also we can control the mechanical properties of the whole nanofibers wound dressing. And here is just some data. So by using different concentration, and by using different cross-linking time, and also by using different degree of the C.C, so we can control the mechanical properties of the JAMA, and we can make these scaffolds very elastic as well. And when we see something on top of the very elastic JAMA, so because when the cells are seated on top of something very soft and elastic, they can sense further, and then they can migrate deeper. So and then we can see that when we see the cells on top of the JAMA, they can migrate deeper, and they can infiltrate the whole like scaffolds. And then we implant these scaffold into the rabbit ear model, and then to see the wound closure. So we can see that both the JAMA and the gelatin, we can see that they can support these kind of the JAMA can support this kind of wound healing, and then we can have the very nice organization of the collagen. And this to show that the gelatin, these JAMA, they can have like tunable physical and biological properties. So while controlling the cross-linking density of the JAMA, so we can control the physical mechanical properties of the materials. While also controlling the changing the mechanical properties of the materials, we can control the biological properties of the materials. And also we can make them into the nanofibers, and also they can be used as wound dressing. And also we make these kind of electrospun fiber scaffolds as the artificial periosteum. Here is like here is like what people now have used the materials. They're using the PLGA, PLLA, or PCL to make these kind of nanofibers scaffold for periosteum. And these materials, they are all synthetic materials, so they might be lack of the bioactive moieties, and also they have limited osteogenesis and angiogenesis potential. And so what we do is like we are again using the JAMA, and also inside JAMA, so we are encapsulating these hairy hydroxyapatite nanoparticles. So this is the hydroxyapatite nanoparticles with C-C. So they can cross-link with themselves, and also they can cross-link with the materials. This will increase the mechanical properties of the materials. And also we use the C modified arginine. So arginine is like, so first of all, the arginine, they are positive charge. So they can have the electrostatic with the tissue, so they can increase the tissue adhesion. And also when these materials, they degrade, they're going to release the calcium and also the arginine. The calcium, what is the function of the calcium? Is like they can increase the nitric oxide synthase, and also the arginine is the precursor of the therapeutic gas, nitric oxide. So with these two elements here, we can increase the production of the nitric oxide gas, and also we can accelerate or activate the signaling pathway to have the both the angiogenesis and osteogenesis. So here, this is again like we're showing that we have successfully synthesized the JAMA and also the Photocrosslinkable L-arginine. And this is the morphology of the electrospun fibers. So we can see that by adding different concentrations of the hydroxyapatite nanoparticles, so we can increase the mechanical properties of the nanofibers scaffolds. And also we can see that our materials is very, they can they have very good tissue adhesion, so they can adhere to bone, and also they are elastic. So this is to show that the biocompatibility of the electrospun fibers. So we can see that when we are adding different concentration of the L-arginine, and also when we are adding different concentration of the hydroxyapatite, so both of all these composite nanofiber scaffolds, they can support these mesenchymal stem cells survival and also adhesion. And also when we're seeding the human endothelial cells on top of these scaffolds, you can see that most of the cells, they are alive, and also they can adhere well to the scaffolds. Now also we can see that when we are increasing the L-arginine concentration, because the cells are negative charge and the L-arginine, they are positive charge. So we are going to increase the charge of the membrane, and then they can have better adhesion to the cells. And then this is to show that the materials, they can support the osteogenesis. So again, we're using two markers. One is the alkaline phosphatase, and the other one is the alizarin red. We can see that our composite materials, when we're adding different L-arginine and also the hydroxyapatite, so we can enhance the angiogenesis of the mesenchymal stem cells. And then we examine the angiogenesis potential using the composite electrospun nanofibers. And again, we can see that when we are adding more arginine and also more hydroxyapatite nanoparticles, these materials can support better angiogenesis. And then we analyze why these materials, they can support better angiogenesis. So this is because when the materials, they degrade, they're going to release the calcium and also the L-arginine. And these two components, they can activate these signaling pathway. And this signaling pathway is what we call our NO-cGMP signaling pathway. They can regulate both the mesenchymal stem cells and also endothelial cells. And then they can enhance the osteogenesis and angiogenesis. And then we apply these materials on the mouse cranial defect model to see if the materials can support the bone regeneration. And then we can see that when we are adding the L-arginine and also when we are adding the calcium, they can support better bone regeneration. And we also examine the materials, how good they can enhance the angiogenesis. And we can see that these red circles, they indicate the blood vessels. And then we can see that we have more blood vessel formation when we're using these composite Electrospun fiber scaffolds. So this is to show that by using these Photocrosslinkable materials, so again, we can tune the mechanical properties of the scaffolds, and also by controlling different components, by just using very simple components, we can activate some cell signaling pathway, and we can achieve coordinated osteogenesis and angiogenesis. And then we're going to talk about how we can use the JAMA hydrogels to make the cell-laden microspheres. So what we do, so why we want to use these microspheres is because when we're using the bulk hydrogel, if we encapsulate the cells in there, so if the materials that are huge, because lack of the diffusion, so the nutrition cannot enter the center of the hydrogels, so the cells in the center may be dead. So in this case, what people develop is like we develop this microstructured cell delivery system so that nutrition can diffuse and then we can have better cell survival. And then the reason why we are using the microfluidics is because they allow one-step fabrication of the large quantity of the cell-laden microspheres. And also this work is like what we have done, what I've done many years ago when I was a postdoc. So nowadays in my lab, so what we're doing, like we're using electrospray, so it's similar techniques of the electrospinning, and we can even like make the large quantity of the cell-laden microspheres in a very fast manner. So here is just like what we can do. So what we have done, what I did before is like we use glass capillary. So here this is a JAMA solution, and outside is the oil. So by forming these water in oil, we can form these kind of droplets. And while you controlling different flow rates, we can control these microsphere sizes. So the one we aim is like 100 and something. And because like when we are having large these large balls, we can encapsulate more cells, and then they can have better function. But when the balls are too large, they will be have the same issue. It's like the nutrition cannot diffuse in, so we might have the cells in the center dead. So in this case, we are achieving to we want to have something like between 100 to 200 microns. And this is to show that the JAMA microspheres, they can support the cell survival and also the cell spreading. So we can see that after like culture for 7 days, so most of the cells are survived inside the JAMA hydrogels. And also we can see that the cells will spread. And also this is something very important. So we can see that when we culture the cells for a long time, so first of all, we can have lots of cells inside the materials. That means that the cell can proliferate inside the JAMA hydrogels. And also we can see that the cells migrate from inside to outside from inside the microspheres to outside the tissue culture plastic. So this is to show that, so if we actually inject the microspheres into the bone defects, the cell encapsulated will migrate out and involve in the bone regeneration process. And also we show that these JAMA hydrogels, they can support the osteogenesis, as we can see that with time, we have more these calcium deposit inside the microspheres. And then what we do is like we inject these cell-laden microspheres into the rapid femoral condyle defect. And with time, we can see that after 4 weeks of the implantation, we can have these more new bone formation. So when we're using the JAMA hydrogel, cell-laden JAMA hydrogels, we're going to have more these cell-laden, more of these kind of new bone formation. So this is to show that these photo-crosslinkable JAMA, they can also be fabricated into the cell-laden microspheres, and also they can support the osteogenesis in vitro and in vivo. These are some ongoing research with the photo-crosslinkable polymers. So we can, for example, this is what we this is PDMA. So the materials, they are injectable, and also they are hydrophobic. So they can cross-linked underwater. So what we do is that we inject them to this is the osteochondral defect, and then we cross-link and to ask the materials to regenerate bone and cartilage. We just got this paper published by AFM, and also we are also using the materials as a microneedle patch. So here, the red color, that's the PDMA, and also these blue color, they are JAMA. So actually, like, although they are different properties, but like because they are photo-cross-linkable, so we can actually make these two materials, they are on top of each other, and their bonding, they are very strong. So by using these microneedles, because the JAMA, they are hydrogel, so when they are these microneedles, they are in the skin, because the hydrogel will absorb water, and then these will further actually fix the microneedles to the skin. And also we're making the materials into a cardiac patch, because we can control the M and N of the propylene glycol and also the lactide. So we can make the materials very elastic. And we're also making them as a cardiac patch. And also we can make the materials sprayable. So we can spray the materials and then to the skin wound, and upon light exposure, the materials can be solidified, and we can have these kind of sprayable wound patch. So these are my students and my research group. So I think these students are very important for the research projects. And I sincerely appreciate my students, and also I sincerely appreciate these funding. So without their support, I won't be able to do I think such excellent research. And thank you very much. And we do welcome collaborations.
Thank you very much, Dr. ZHAO. That was a very, very exciting talk. And from a photo-cross-linkable polymer, you generate so many things. I'm pretty sure the audience will have a lot of questions. So maybe I invite audience, online ones. If we have a question, you can raise your hands for asking some questions.
Hello, Dr. ZHAO. May I ask a question?
Sure, go ahead.
Can you identify yourself?
I'm Zhang ZHAO, and work on the PLA oligomer for antimicrobial material. And thanks for your explained talk. And I would like to know, is there any chance to use the PLA oligomer in your system, such as wound patch or other Electrospun fiber or other system?
Yes. So I have collaborated with Professor Tao using your materials. So what we do is that we make the materials because your materials, they are antibacterial. So what we do is like we make them a coating, and then and also we give some other lubricating properties. And so these so in this case, we can reduce the bacteria by either killing the bacteria, and also like by lubricating the bacteria. So in this, we can make these antibacterial coatings. For other things, I'm actually still figuring out for the PLA for that one.
Yeah, PLA, if the polymerization degree is enhanced, it also can be solid. And can it be used in the bone or other organ or tissue?
PLA already been used in the bone. So because like PL, so if you look at the materials we are making, so we also modify them from polylactide. So because the polylactide, they are degradable and also they are very strong. So and in this case, we can make them for bone regeneration.
Oh, okay. One last question. It's about the gel mat. You have been commonly used for the healing the bone. Can it be absorbed by the tissue or continuously exist in the body?
Yeah, for the gel mat, because you can see that the major component is the gelatin. So what they can do is they can be degraded by the cells. The remaining is going to be like the mesacrylate groups. So those things will be like excreted by the by the liver and then to the urine. So because those are tiny small molecules.
Okay, thanks for your answer. Thanks for your talk.
Okay, thank you. Any more questions? Maybe I ask a question. Dr. ZHAO, I'm very interested to listen to your talk, especially the progress made in this use of this polymer in tissue engineering. Many years ago, I remember people used the fiber structure to make a scaffold. And one, you mentioned that there are some problem with two-dimensional, those electrospinning fabric cannot allow the cell penetrate. That is very good comment, I think. The second one is in those days, I remember somebody said during the tissue growth, people may apply certain strength. That means elongation in one direction and repeatedly, cyclically. That will generate some cell orientation or even moderation of the mechanical properties of the cell. What is the progress in that area? And can you comment on it?
So what we say is like it's not the Electrospun fiber. So now, so if you just like, for example, I don't know how to go to the first one. So if we are just using the Electrospun fibers, if for example, if we just electrospin these PLLA, because the materials itself is very stiff, and also when we are making the fibrous layer very thick. So what we end up is like the pore size is very tiny, and then the cell cannot penetrate. But there are actually many ways to change this. For example, what people do is like we can make these kind of fibers. So what they do is like what people have done is like after the electrospin, for example, PLGA, they put them in the ethanol and then they dry them. So after the immersion of these materials in the ethanol, this fiber is no longer straight, become like spiral shape. Yeah, they become like spiral shape. In this case, we can make the materials like elastic. So this is one thing. And the other one is like also we can actually change the way how we can electrospin the materials. For example, so nowadays people also make the offsetting material, more offsetting structure using the electrospun fibers. So in this case, by varying also like, so we can actually change the pore shape of the electrospun fibers. And also in this case, we can make the materials elastic. So in this case, for example, when we are making the electrospun fibers as a skin wound patch, so because in this case, they are stretchable, right? So they can fit the skin better compared to these. This is a very conventional way of electrospin fibers. So these are still very, I wouldn't say that, well, I would say that electrospinning is still very popular, and also like these kind of wound dressings, they are very popular research, and they get a lot of these kind of like investments. So a lot of companies actually give money to do this kind of research. It's just like the way we are now electrospinning is like keep changing, and there are many other ways to modify the materials and also modify the structure, and we give like different properties of the fibers. And just the way I'm using them is like I make the materials itself, they are elastic. So we can make the Electrospun fibers elastic. But there are also very other ways, just by changing the pore shape, or just like immersing them in the ethanol. They can also, they change the shape of the fiber, or they can change the shape of the pores, and then they can change the mechanical properties of the wound dressing.
Yes, thank you. Another question I have is, it seems to me you have achieved to create some kind of network from maybe tens of angstrom to maybe few micrometer, kind of a range. Any gap in between? Because by cross-linking, you can get a very tiny kind of cross-linking, right? Then you jumped into printing, 3D printing, or this microfiber. That is in the order of a micrometer. Then somewhere in the middle, say 100 or below 100 nanometer. Anything there? Any technique we can create, kind of a cross-linking or some kind of structure?
Oh, this is just the this is just how we can use the photo-crosslinkable materials. So because the materials itself, they are liquid, and also we can change the viscosity of the material. So because when we are changing the viscosity of the materials, so for example, we can make them very like watery. So in this case, we can spray them. And also, so when they are watery, and also, so in this case is, yeah, so we can spray them. And also by changing the viscosity, we can electrospin them, and also we can 3D print them. So this is just the, I think the nature of the photo-crosslinkable materials itself. And then, so because they are initially liquid, and we can change the viscosity, and also we can change the, to make them hydrophobic and hydrophilic. So we can actually give a lot of different applications of the materials. And also one thing I didn't mention, because I'm just like recently start working on this, is that we try to give the so one thing I didn't mention here is like we can actually control the gradient scaffolds. And also like somewhere in the scaffold, we can make them like stiffer, and also by controlling the cross-linking density. And somewhere we can make them like softer. So in this case, we can make these kind of gradient scaffolds. So for example, to treat these kind of, for example, tendon to bone or these kind of joint. So because they are, so we need to match different mechanical properties.
Yeah, that sounds exciting, actually. Application of this would be much wider than tissue engineering in many other fields, and probably people are interested to make a gradient cross-linking.
Yeah, and also once we make the, I'll just like to talk about, so just talk about the Electrospun fibers, and also like in addition to wound dressing, definitely there are many other application, like we can make them like a filter, and also the same thing, we just change the fiber structure and also change the pore structure. We can give a lot of different physical properties to the scaffold, and we can have many different applications in biomedical applications, and also in the more like industry applications, or like engineering for our daily life as well.
Yeah, the material itself has a beauty of easy processing and using conventional method. So I think the application scope is very wide. Yeah.
Thank you.